Hello my dear aspirants, I hope you all are doing well. So I am again back with another topic for you, which is the transformation principle by Griffith's experiment. This topic is needed in your class 12, Neet, CSIR NET, SLET and all life science examinations. So I hope this topic will be helpful enough. Watch the video till end, and if you don't understand any particular point, you can comment down below. So let's start the video. So the topic for today's class is Griffith's experiment, which was performed by Frederick Griffith and it was reported in 1928. Before this experiment, it was not known that DNA is the genetic material, and the concept of genetic material that can get transferred from one organism to another was not known till this experiment. This experiment actually showed the transformation principle. That means the genetic material from one bacteria can be transferred into another bacteria with the help of a process called transformation. That thing was proved by Griffith in his experiment. So the experiment suggested that bacteria are capable of transferring genetic information by a process called transformation. That means there is something called genetic material, and that genetic material can be transformed from one organism to another organism, and later it was known that genetic material was DNA. But this experiment is only telling us that the genetic material can be transferred from one organism to another, but it was also not known that genetic material is DNA, which will be known in other later experiments. But now we will only discuss about the Griffith experiment. So in order to do the experiment, he took two strains of pneumococcus bacteria. Okay, there are two strains which were used. One is the smooth strain, which is virulent, that means that can cause disease. Another is the rough strain, which is non-virulent, that means it cannot cause disease. Fine. So he took the rough strain first, and that rough strain was injected into the mouse. Okay, then what should be the outcome? Because rough strain is non-virulent, it cannot cause any disease, or it will not kill the mouse. So this rough strain, if it gets injected into the mouse, then the mouse will live or the mouse will die. Definitely the mouse will be alive, right? Because it is non-virulent, it cannot kill the mouse. Then again he took smooth strain of the pneumococcus, and then he injected into the mouse, and it was seen that the mouse died. Of course the mouse will die because it is the virulent strain. Later again what he did, he heat killed the smooth strain, and when the smooth strain of bacteria will be heat killed, then it will not be able to cause or it will not be able to kill the mouse. Fine. So the mouse will be alive or the mouse will live because it is heat killed. Although the smooth strain is present, but it is heat killed. It is inactivated now. But when he mixed the rough strain is also non-virulent, heat killed smooth strain is also non-virulent. But when these two strains are mixed, then surprisingly it was seen that but this mouse, it should be alive now because both are non-virulent, but surprisingly it was seen that the mouse died. Okay, why the mouse died in this particular experiment? Because this particular experiment has shown us that transformation happened. Imagine the smooth strain were heat killed, okay, but the DNA, that means the genetic material, was still there in the particular surrounding. Okay, this genetic material was still there in the particular surrounding. And what does transformation actually mean? Transformation means that taking the genetic material from the surrounding by another bacteria. This smooth strain will release the DNA, that means the genetic material, and this genetic material, when they are both of them are mixed, then this rough strain will take up the genetic material from the smooth strain. Okay, it will take up the genetic material from the smooth strain. That means in simple terms we can say that the genetic material from the smooth strain will get transferred to the rough strain. As a result, the rough strain will now become virulent because all the information are present in the genetic material only. It can be DNA, RNA for different species, but let's take example that it's DNA only. So all the information will be present in the DNA. That means the capacity of virulence, okay, the virulent property is also present in the DNA. So when the rough strain will take the DNA of the smooth strain, then the rough strain will become virulent. As a result, this rough strain, will now become virulent because it is taking the genetic material from the smooth strain. Although the smooth strain were heat killed, but the DNA was not killed. DNA was still there in the surrounding, which was taken up by the rough strain. As a result, the mouse will die. This has proved that transformation was happening. Transformation was happening means transfer of genetic material from the smooth strain was happening into the rough strain. When transfer of genetic material from one organism into another organism has taken place, that means it is called transformation in case of bacteria. I hope this transformation principle is clear with Grifith experiment. First rough strain is injected, then the mouse will not die. Then smooth strain injected, the mouse will die. If the smooth strain are heat killed, then the mouse will not die because now the smooth strain has no property of virulence. Then what will happen if the rough strain and the heat killed smooth strain are mixed, because genetic material will be there in the surrounding, only the smooth strain bacteria is heat killed, it is inactivated. Now the mouse will die because now the rough strain is taking the DNA of the smooth strain and now it will become virulent. I hope this is clear, but till this experiment, remember this experiment is not able to tell us that the genetic material is DNA or RNA or protein or whatever. In the later experiments, it becomes clear that the genetic material is DNA. But here, at least we know that something is getting transferred from one organism to another organism. If we elaborate this concept, that means the hereditary, that means the transfer of genetic material from parents to offspring. This concept also came after this experiment only. I hope it is clear. I just want to provide an additional information that if you want to take detailed coaching of the SLET examination, then you can comment down below or you can contact in the given number. I provide full course or full coaching of the SLET Northeast SLET exam, that means life science. So you can just comment down below or you can contact in the given number. And if you like my videos, then please don't forget to subscribe the channel and you please share the videos with your friends.